I'm Kevin Kavanagh from HealthWatch USA. HealthWatch USA is a patient advocacy healthcare policy research organization which was founded over 10 years ago. I'd like to speak to you today about the importance of nurse empowerment with the theme, "It is not the nurse's fault." Nurses face the daunting task of safeguarding the health, healthcare, and safety of both themselves and their patients. And I feel there's no better testament to this problem than the recent Ebola outbreak, when it was initially stated that any hospital could take care of Ebola patients. At the same time, HealthWatch USA and a Lexington Herald Leader op-ed was calling for better standardization of infectious disease practices and a need to take dangerous pathogens more seriously. And then Nina Pham, a nurse, became infected. Initially, she was blamed for her own infection. However, it was soon learned that there were ineffective standards with lack of appropriate PPE at her facility. A fully hooded hazmat suit and the buddy system were not available. Since then, steps have been taken to emphasize containment and control of dangerous pathogens. In June of this year, the CDC reaffirmed that isolation of carriers and those infected with MRSA is key to controlling the spread of this dangerous pathogen. However, I feel that many of our protocols to safeguard healthcare workers and patients from MRSA are based on a misinterpretation of the literature and two poorly designed studies. In 2003, European researchers Cooper et al. Found that most studies supporting surveillance and isolation of MRSA carriers were before and after studies. However, despite this, they recommended staying the course. The United States, however, decided to veer off of course and abandon, or all but abandon, this time-tested intervention. And they then backed up this decision of not performing surveillance for MRSA carriers with two poorly designed studies. The first was Harbreth et al., which was published in JAMA, where appropriate MRSA prophylaxis was not given to the majority of pre-surgical patients that were in the intervention group. And the second was Huskins et al. In New England Journal of Medicine, where it took 5 days to get back results and start patient isolation procedures. And contact precautions in this study were not strictly followed. The United Kingdom instituted a hand hygiene campaign and mandatory and screening isolation for MRSA carriers and saw a phenomenal decrease in infections. This figure illustrates the percentage of MRSA and staph cultures in the countries in Europe. The United Kingdom is the purple line. They saw a 60% decrease in methicillin resistance in Staph aureus cultures, along with a concomitant decrease in MRSA infections. But they did not observe a decrease in MSSA, methicillin-sensitive Staph aureus, or E. coli bloodstream infections. Hand hygiene was performed across the board and should have impacted all pathogens, but only MRSA, which had implementation of screening for carriers, showed a decrease in infections. So there's no guarantee that hand hygiene will work as the main intervention. It must be a bundled approach. And in the context of multi-drug resistant organisms, hand hygiene is extremely important. But it is a backup measure. These organisms should not be on a healthcare worker's hands in the first place, and if they are, there is a problem with containment and control. This graph shows data which HealthWatch USA published in Antimicrobial Resistance and Infection Control. The orange line is derived from NHSN data and shows an increase in severe MRSA or bloodstream infections in 2015 in the US private sector hospitals. The blue line shows a huge decrease in VA hospitals. The Veterans Administration screens all admissions for MRSA. However, the two baselines are not comparable. They both use different definitions of infections, and they both have risk adjustments, which are different. In the 2015 increase, it was found by the CDC that it may have been due to how the United States accounts for community MRSA. So we then learned that hospital rates are adjusted for community rates. They're also adjusted for being a teaching institution and bed size. Thus, rates of infection and risk adjustment are more for hospital performance and accountability than they are for looking at the risks of workers or patients of acquiring the infection. Healthcare policy workers then focused on the reduced MSA study and daily chlorhexidine bathing. This is bathing every patient in the unit every day in chlorhexidine. Now, I have a number of significant concerns which we've published regarding the reduced MSA study. First, there is some confusion that the study showed that MSA screening and isolation wasn't effective. This comes from Group 1, where they said they implemented screening, but in fact, both arms of Group 1 had screening and isolation. And this group was to control for trends over time. It wasn't to evaluate the effectiveness of this intervention. It also used surrogate endpoints that of primary objective was clinical isolates as opposed to infections. But most importantly is that there were significant changes in metrics. After the trial completion date, the metric for all pathogen bloodstream infections was added. And in the abstract, it states universal decolonization was more effective than targeted decolonization of screening and isolation in reducing rates of bloodstream infections from any pathogen. I've heard federal policy workers testify to this before the Presidential Committee on Infectious Disease. But what's not stated is that the all pathogen bloodstream category is a composite category composed of many bacteria. MRSA bloodstream infections, their decrease did not reach statistical significance. And by far, the greatest decrease was in yeast and commensal bacteria, such as Staph epi. HealthWatch USA then participated with the Reuters investigative report, and I'll only mention some of their findings. But overall, they found that a well-known researcher and author of the reduced MSA study had at least the appearance of a significant conflict of interest with the manufacturer of the chlorhexidine product which was being evaluated. And this was to a tune of $1 million, which was earmarked to this author's research fund by a foundation which was connected to the manufacturer of the product that was tested. In addition, this investigative report found that this type of usage of chlorhexidine was not FDA approved, and they also found an FDA warning against using chlorhexidine cloths as a general skin cleanser. For their efforts, this investigative team was awarded First Place Journalism Award by the American Association for the Advancement of Science, and this had to do with the tracking of MRSA infections. This association is very prestigious, and it publishes the journal Science. Later that year, the LA Times reported that at the lead author's facility, there was an MRSA outbreak which was not controlled with chlorhexidine bathing. And at Infectious Disease Week, the first author of the reduced MSA study reported that in general units outside of the ICU, only patients with central lines and other medical devices derived a benefit from chlorhexidine bathing. Now, in addition, I would like to add that chlorhexidine is an extremely complex chemical molecule. It has 22 carbon atoms. So it's similar to an antibiotic, but it's defined as an antiseptic because it has such a wide spectrum. And you can get resistance to chlorhexidine. It can induce resistance to other antibiotics. And there is even evidence that it can induce resistance to the last line of defense antibiotic, colistin. And some articles have indicated that it can be one of the drivers of the CRE epidemic. So what I have seen is that many people in the lay public, the lay press, other allied fields of science, and also in foreign policymakers are concerned about the direction of infectious disease policy in the United States. For example, the World Healthcare Organization recommends screening and decolonization of all pre-surgical patients for Staph aureus. In the United States, we do not even routinely do this for MRSA. So we must have better nurse staffing and nurse training. We need to support our nurses. They need to have time to do proper hand hygiene. They need to have training and time to do proper taking on and off of protective gear. And despite what you've heard, I do not believe that isolation causes an increase in adverse events or depression if you have proper nurse staffing at a facility. The bug study, which universal glove and gowning, published in JAMA, also concluded this, that there was no difference in adverse events in patients that were isolated, and they also found a lower risk of MRSA acquisition. Very good prospective double-blind study, and this confirms the Northwestern study regarding screening and isolation of MRSA carriers. So I feel that on a system level, this has led to the adoption of a one-size-doesn't-fit-all approach, and kind of facilities need to tailor what's needed for them. And in my view, sometimes this leads to the easiest way out or purchasing the cheapest set of shoes. No one's told me why one facility size is different from another. And certainly, at points, a lackadaisical attitude towards some of these dangerous pathogens. And we saw this in the Ebola epidemic. And not making the very large and needed investments in staff training, and equipment that are needed to stop this epidemic. There's a lot of talk about how we need to do this, but to get someone to hire an infectious disease nurse takes almost an act of the Almighty. We need to put our resources where our thoughts are. So in conclusion, I'm reminded of an old Chinese proverb: if we do not change our direction, we are likely to end up where we are headed. Thank you.